Hello everyone. Welcome to today's live broadcast, How Anthropogenic Chemicals Are Altering the Microbial World. I am Brenda Kelly Kim of LabRoots, and I'll be your moderator for today's event. LabRoots.com is the leading scientific social networking site, and we are proud to bring you this interactive web seminar. For more information, please visit us at LabRoots.com. Here's how the presentation works. We want to hear from you. Questions, comments, and even answers can be submitted via the green Q&A button at the lower left of your screen. We'll try to get to everyone, but if not, we'll make sure to follow up with you via email. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you can't hear or see this presentation properly, let us know by clicking on the support button at the top right or the Q&A button in the lower left. I would now like to introduce today's speaker, Dr. Erica Marie Hartmann. She is an environmental microbiologist interested in the interaction between anthropogenic chemicals and microbial communities. She was the first graduate of the Interdisciplinary Biological Design PhD program at Arizona State University, where she worked with environmental engineers to develop molecular methods to detect microbes in food, soil, and water samples. From there, she was awarded a Fulbright to study microbes that degrade the toxic carcinogenic pollutants known as dioxins in France at the Commission for Atomic Energy. In her current work at the Biology and the Built Environment Center at the University of Oregon, Erica is leading studies on the effects of antimicrobial chemicals on the microbes found in indoor dust. In addition to her research, Erica serves on the American Society for Microbiology's Microbiome Communication Task Force and as a University of Oregon expert in microbiology. Let's turn it over to Erica now.
Great, thank you. So to start out with, I have a CME disclosure, and for those who are interested, it's here. And with that, I'll just get started. So yeah, I'm going to be talking about how anthropogenic chemicals, which are chemicals that we humans make, are altering the world and the way that microbes make their living. And the first thing that we need to all accept here is that we humans introduce chemicals into the environment, and we can do that intentionally or unintentionally. And once these chemicals are in the environment, they interact with microbes that we did or didn't know were there. And the thing about microbes is that they're so much more than just their 16S genes. And one thing that we tend to do when we're looking at microbes in an environment is focus just on looking at who's there. But I like to think of microbes as bags of enzymes. So they're proteins, and they're responding to the physical and chemical stimuli in their environment. So bacteria, microbes, like all cells, are made up of 4 major macromolecules: lipids, carbohydrates, nucleic acids, and proteins. And we care about these macromolecules because they're common among all cells, but also because they do everything that the cell does. So that can be energy storage or structural support, protection, catalysis, transport, defense, regulation, movement, heredity, and information transfer. These are all things that most cells do, and all of these actions are actually performed by the macromolecules in the cell. When we look at the breakdown of macromolecules in a cell, most cells contain a majority of proteins in addition to the nucleic acids that form the genetic information, the hereditary information. And then there are carbohydrates and lipids. And one thing that I'm going to talk a lot about is proteins and nucleic acids, because those are the most informative macromolecules. And proteins especially I find really interesting because they're what define the functions of the cell. So bacteria perform many functions, and all of these functions are regulated by proteins. So for example, digestion in your intestines. If you know anything about the human gut microbiome, you know that one of the things that it does is it breaks down food that our human cells can't metabolize. But microbes also perform a lot of ecosystem services like nitrogen fixation. They digest sludge in our wastewater treatment plants. They change the oxidation state of metals in the soil, causing metal leaching. They can be used for bioremediation, so cleaning up pollutants that we spilled in the environment. But proteins can also be virulence factors or efflux pumps. And these are two things that we're going to come back to. But basically, proteins do everything that the cell is doing. All of those functions are linked to proteins. And so you need the genetic information to code for these proteins. But that genetic information, while it'll change a little bit, certainly more in bacteria than it does in eukaryotes, for example, in humans, what's going to change much more dramatically over time is not the genetic information itself, but whether or not a gene is expressed. So if we look at over the course of development of a human, for example, even though the genetic information doesn't change, the protein expression changes quite dramatically, and that's how we develop from fetuses into humans. So much like a developing multicellular organism, we can think of a microbial community changing over time. And the big difference is that the genetic information will change more in a microbial community than it would in a eukaryotic organism. But in addition to changes in genetic information, and that can be through sharing genes or just changes in the relative abundance of different species, we can also see changes in the phenotypes of different species. And even within a single species, changes in phenotypes of individual cells. So what that means is that the proteins that these cells are expressing are going to change over time. And as we'll see, they're going to change in response to chemicals, especially chemicals that we humans make. And I'm going to go through a really detailed example, bear with me, just so you can get in the sort of mindset of how bacteria think. And this is going to help us think later about how bacteria might respond in different situations. So we're going to start out looking at E. coli and looking at the growth curve of E. coli. So this is the textbook growth curve when you start a culture, a batch culture. You typically have this phase in the beginning called lag phase where there's not a lot going on. And then all of a sudden, the cells start growing like gangbusters in log or exponential phase. And then they hit some sort of limiting situation where they go into stationary phase. So maybe they ran out of food or there's not enough oxygen or whatever happened. And then slowly the cells start to die off, the death phase. And the traditional explanation for this lag phase, the phase at the beginning where there's not a lot going on, is that the cells are adapting from one phenotype to another. So they're expressing one set of proteins that's not perfect for growing in that culture, and they move towards expressing another set of proteins where they are growing well in that culture. And so in this diagram, that first state, the not super well adapted state, is shown in yellow, and it moves over time towards the purple well adapted state. But it turns out that that's not entirely correct. And so there are a couple of different explanations for what causes lag phase if that is not the explanation. So you can see two here, and it doesn't really matter for the purposes of our example which one's correct. But the basic idea is that you start out with a non-adapted population, that yellow population still. And then either there's a very small fraction of the population that is well adapted, and that's the fraction that's going to take off. So we can see that purple curve going up on the left. Or a certain portion of the non-adapted cells will diversify. They'll actually change phenotype. Some of them will and some of them won't. And the ones that do, that adapt really well, again, those are the purple ones, the ones that take off. And so basically the idea, the take-home message, is that we have multiple phenotypes within the same population. We have purple growing cells and yellow non-growing cells. So to look at how this works, we're going to go through one example of shifting E. coli from glucose to acetate. And the basic experimental method behind this is to take E. coli that's growing on LB, so in plates, rich media, E. coli are super happy, pick colonies and transfer them into minimal media containing glucose. E. coli likes glucose, everybody's growing, very happy. Transfer them again to glucose, so everybody's really, really happy going on glucose, everyone's well adapted. And then in the middle of the exponential growth phase, transfer them from glucose to acetate, which is a gluconeogenic substrate. And we'll see what that does to the phenotypes of the cells in the population. And what we're really interested in here is looking at the shift between glycolysis and gluconeogenesis. So the idea is that when cells are growing on glucose, they're using the glycolytic pathway, which is this one here. And when they're growing on acetate, they're using the gluconeogenic pathway, which is basically the same pathway but run in reverse. And either way, both of these pathways take carbon from outside the cell, glucose or acetate, and funnel it into the TCA cycle so that the cell can make all of the things that it needs for carbon and generate energy so that it can do things. And if we look now at our population of cells that were grown on glucose and then shifted to acetate, we can see that there are two curves here. So this is looking at fluorescence of the cells themselves. And in the membranes, some of them are actively dividing, which is going to change the way they fluoresce. So you get these two curves. One, the yellow curve, doesn't change a whole lot. This is the fraction of cells that are not growing. So that's our phenotype number 1. And phenotype number 2 is the purple cells, and those are the ones that are actively growing. And if you look at the graph on the bottom, you can see that the yellow cells are not really changing over time, whereas the purple cells are growing up, which, if you look at them together, is what gives us our textbook lag phase growth curve. So again, textbook lag phase, these are all the same cells, but we have two phenotypes, one that's growing and one that isn't. And we know that this is in response to a chemical stimulus, acetate, because if we increase the acetate concentration, we directly influence the phenotype. We can change the growth rate, as we see in diagram A here. And in diagram B, we see that we can also change the fraction of cells that are actively growing. So the proportion of the population that goes into phenotype, the growing phenotype. And this makes sense. So basically what happens is that if we take away glucose, we're favoring growth on acetate. But only some cells do this, right? So we have these two populations. Even though all of these E. coli have the same genetic potential, only some of them are going into this gluconeogenic pathway. Why is that? Well, some cells can immediately use acetate as soon as that glucose is gone. And this may be due to stochastic differences in gluconeogenic enzyme expression. So basically what that means is that some of these cells, just by chance, because this is a dynamic system, had a little bit more of the proteins that they needed to use acetate right when that acetate was available. Also, the gluconeogenic pathway in E. coli is regulated by post-translational protein modifications. So again, maybe just by chance, some cells had a little bit more or a little bit less of these post-translational modifications. So as soon as that acetate's available and as soon as the glucose is gone, these are the ones that take off. Everybody else, they don't make it, but they don't actually die. And what happens to them is really interesting. So the non-growing cells are not actually dead. They are dormant persisters. And we can see this if we take the cells after they've now been grown on acetate and they're not actually growing. We transfer them back to rich media. Everybody's happy on rich media. And we see that 100% of the cells recover. So what we're looking at is viability over time. And what's interesting is if before we put them on rich media, we treat them with an antibiotic, we treat them with kanamycin, which only affects actively growing cells, you still recover 100% of your population, more or less. However, if you treat them with afloxacin, which is an antibiotic that attacks all cells, whether they're growing or not, you see a much reduced recovery. So this is log scale. We're seeing from about 100% down to about 20% of viability. So that's really interesting that just by changing from acetate or from glucose to acetate, we've created antibiotic resistant bacteria. So I'm going to give you my conclusions about what we just went through. You feel free to draw your own, but my conclusions are that even in a genetically homogeneous population, microbes can be phenotypically diverse. So even with the same set of genetic potential, the same set of genes, we can have different phenotypes. And even though we have different phenotypes, we can have different levels of metabolic activity, but the same viability. So even if a cell isn't doing anything right now, that doesn't mean that it can't be doing something in the future. And also, within the same population, individual cells can have different antibiotic sensitivities. So the cells that were actively growing were sensitive to kanamycin, and the ones that weren't were not sensitive. And these differences arise due to external chemical signals. So in this case, we were talking about glucose and acetate, but we're going to talk more about anthropogenic chemicals, so chemicals that we humans make. So one set of chemicals that I'm really interested in is antibiotics. And we're going to start by talking about antibiotics and the human microbiome. So humans introduce all kinds of chemicals into our bodies. Things like glucose and acetate might come from our food or metabolites of the things that we're digesting. So you eat a hamburger or whatever it is you're eating, that's going to be broken down into smaller macromolecules that the bacteria in your gut are going to see and start digesting. But we also ingest bioactive chemicals that we call drugs, some of which are targeted specifically at microbes, and these are what we call antibiotics. And when we ingest drugs, basically it's just sending different chemical signals down to the microbes in our gut. And the human microbiome, especially the gut microbiome, performs lots of different services for us as hosts. So the microbes in our gut, for example, digest our food and keep our immune systems working. They're going to take those chemicals, the chemicals from the food that we're eating, and some of the proteins, for example, that we can't digest, they will digest. They're going to make signals that are passed to the host immune system and basically keep everybody happy and healthy. But we also expose them to antibiotics. And these antibiotics, I'm going to talk about antibiotics as if they're one lump thing. But in fact, of course, there are many different classes of antibiotics, and different antibiotics have different mechanisms of action, and they're going to affect more gram-positive versus gram-negative bacteria. They're going to have wider or narrower spectrum. But I'm going to talk about them as if they all have one mechanism of action, or not one mechanism of action, but one effect. And that effect is primarily changing the microbial community composition. So the idea is that we take antibiotics, and before antibiotic treatment, there's one set of microbes in that community. And some of them, just by chance, just like the microbes that could or could not deal with acetate, can or cannot deal with the antibiotics that we expose them to. And so after antibiotic treatment, you're going to see a change in the population structure, a change in the membership of that microbial community. And in addition to that, we're actually inducing antibiotic resistance. So the idea is that the microbes that can deal with these antibiotics, the ones that have the antibiotic resistance genes and are already expressing antibiotic resistance proteins, are more likely to survive. So those are the ones that we're going to find afterwards. But then some of those genes are transferable. So maybe even some of the bacteria that didn't have that capability in the beginning can acquire it in the face of this chemical stress. And they do this by sharing antibiotic resistance genes. So antibiotic resistance genes are typically on mobile genetic elements, and they can be shared through events like conjugation or transduction. So conjugation is when two bacteria get together and they share a plasmid, for example. And transduction is the same idea of mediated genetic transfer through bacteriophage. And all of these events increase when bacteria are exposed to antibiotics. So we can look at the spread of antibiotic resistance genes, not just within a single microbial community, but actually throughout the world. So this study is looking at the spread of the antibiotic resistome, the set of all of the known antibiotic resistance genes in the microbiomes of people in various countries. So we've got China, Malawi, the US, and Venezuela represented here. And you can see that in different places throughout the world, you actually have different antibiotic resistance traits. And this may or may not have to do with the chemicals that these microbes are exposed to. So different countries, we have different practices involving antibiotic consumption, and that leads to differences in the patterns of antibiotic resistance genes that we observe. And from these kinds of data, we can actually track those resistance genes back to the original microbes that contain them. And that's really interesting because the same resistance gene does not necessarily do the exact same thing in all microbes. So I just want to take a minute and go discuss a specific case of what may or may not be antibiotic resistance, remembering that these are just proteins, and proteins are expressed in response to specific chemicals. So antibiotic resistance, an antibiotic resistance gene is one that confers antibiotic resistance in a specific situation. But that same gene in a different situation may do something completely different. And this is the case for the 2'N acetyltransferase, which acetylates peptidoglycan. So we can see peptidoglycan here. And what this protein does is it just adds a little acetyl group onto it. This is not antibiotic resistance. And its natural host, Providencia stewardii, is not a human pathogen. So it doesn't encounter, for example, a lot of the antibiotics that we humans make. However, this gene can be transferred to other pathogenic bacteria. And what happens then is they see the antibiotic gentamicin, if we take gentamicin, and this same protein recognizes gentamicin the same way it would recognize peptidoglycan and adds an acetyl group. So now it becomes antibiotic resistance. So my conclusions about the human microbiome is that, of course, there are many different bacteria, but they're all reacting in different ways to the chemicals to which we expose them. So they're going to react differently to the chemicals in our diet, but especially to the antibiotics that we are using to treat different infections. And these bacteria can share antibiotic resistance. So not necessarily the way we might think. Not all of these traits do exactly the same thing in all hosts or in all bacterial hosts, but there's certainly a certain amount of sharing of genetic information that goes on, and this is in a direct response to the chemicals that we are exposing these bacteria to. So once we've taken our antibiotics, they don't actually stay in us. They can pass into the environment where they affect environmental microbes. So this happens, let's say you take an antibiotic drug, a certain amount of that is going to get metabolized in your system, but the rest of it is actually going to pass through and go into the wastewater treatment plant. Similarly, we use a lot of antibiotics in agriculture and especially in raising livestock. And so again, a lot of those antibiotics, some of them are going into the actual livestock, but some of them are passing through and directly through the manure get into the environment. And so what happens to these drugs, these chemicals in the environment, and now they're influencing lots of different microbes that we didn't really intend for them to influence. So a quick note, because I'm going to switch from talking about antibiotics to antimicrobials, and you've probably heard both of these words before. And just by convention, antibiotics tend to refer to medicines for humans or animals. So these are going to be, the drugs that you take a certain number of pills for a certain number of days. And they're really high. We tend to prescribe them in high dose, high intensity for short periods of time. Antimicrobials are agents that are used to disinfect surfaces and eliminate bacteria. So these are going to be things like your antibacterial soaps. And from the bacteria's perspective, these are both chemicals that are trying to kill them. So they don't really see a whole lot of difference. But one of the big differences between antibiotics and antimicrobials is especially the way they behave in the environment, because antimicrobials leave long-acting residues, especially on surfaces, to be disinfected. That's what we design them for, that you apply something and you leave it there and it retains its disinfecting power, as opposed to a drug where you take a certain dose and then when it's over, it's gone. And so these antimicrobials can have a prolonged action. These are referred to as residue-producing antimicrobials, and some really common examples are triclosan and triclocarban. We're going to talk more about triclosan. But so just to say that antibiotics and antimicrobials are just classes of bioactive chemicals that kill bacteria. So triclosan is used in toothpaste and disinfectant solutions, so cleaning solutions and hand wash. These are your antibacterial hand soaps in most households, at least in the US. And from those households, so if you're using a toothpaste or a hand wash, you wash your hands, you brush your teeth, everything is washed down the drain. From there, it goes into a wastewater treatment plant. And in wastewater treatment plant, things are separated into the liquid and the solid components. So the liquid effluent goes out into surface water, and the solids can be applied to soil. From the soil and the surface water, whatever chemicals go along with those effluents go into the groundwater, where they can then make it back into the waterworks and back into our drinking water. And so triclosan is present in consumer products in the milligram per gram level. And when we look elsewhere in the environment, we find them at much lower concentrations, which on the one hand is reassuring, but on the other hand is a little disquieting. Because basically what happens is you have these very long-lived bioactive chemicals that at high doses are lethal, but at low doses, when they stick around for a long time, we might just be giving the bacteria extra time to adapt to be able to deal with these. So that's kind of a tricky thing to deal with, is that now you have these long-lived chemicals that, yes, they're dilute, but maybe because they're dilute, they lose a little bit of their action and the bacteria get a little bit better dealing with them. And one of the ways they get better at dealing with them is by developing multi-drug resistance. So multi-drug resistance comes in a few different flavors. One is the accumulation of multiple resistance genes, each specific to a different drug. So if we remember our classes of antibiotics, I talk about antibiotics and antibiotic resistance as if there's just one thing. But there's a whole host of classes of antibiotics, and there's a whole host of classes of antibiotic resistance genes. So you can accumulate multiple resistance genes that are specific to multiple antibiotics, or you can basically just beef up your overall defenses. So this can be outer membrane impermeability by increasing the concentration of branched chain fatty acids. And this basically just makes the membrane a little bit trickier for anything to get through. So antibiotics outside, we don't really care. Antibiotics in the cell, that's when we get worried. As long as the antibiotics can't get in the cell, we're okay. And these are non-specific. So this is going to be against lots of different classes of antibiotics. Another thing that we can do is have non-specific efflux pumps. So remember, we talked about these earlier. These are proteins that will just indiscriminately pump antibiotics out of the cell. So an example of this are the MEX genes. And these here, your antibiotics get in, and then we immediately pump them out. And as long as we keep pumping them out, everything's okay. And then another option is to go into a persister state. So this is, for example, what we saw in response to the switch from glucose to acetate. That some of those cells, instead of growing on acetate, they just go to sleep. And when they're asleep, they're totally immune to any antibiotics that affect growing cells because they're not actively growing. So the thing about triclosan is that it can actually induce resistance not to itself, but to other antibiotics. So what we're looking at here is different pure cultured bacteria that have been pre-incubated with triclosan and then exposed to either chloramphenicol or tetracycline. And what you can see is that the minimum inhibitory concentration, so that's what we're looking at on the y-axis here, goes up sometimes by a factor of 5. So you need 5 times more antibiotic to kill these bugs now than you did before they were exposed to triclosan. And this happens at environmentally relevant concentrations. So these bacteria were exposed to concentrations that you can see on the left, these 2 left columns showing the different strains of bacteria that were pre-treated with triclosan. And then you can see the concentrations like I showed in the cycle of triclosan in the environment, showing what concentrations we would find in biosolids, sediment, soil, wastewater treatment effluents, groundwater, and surface water. And the effect that we're seeing in these pure cultures is not totally unreasonable given the concentrations that we see in the environment. And we can see not just in pure cultures that we're playing with in the lab, but in complex environmental microbial communities, such as here we're looking at a community from a wastewater treatment plant. And we're looking at MEX-B, that's that multi-drug efflux pump. And if we don't do anything to the system, we can see that the antibiotic resistance doesn't change over time. But if we add triclosan to the system, we perturb the system, in just over 2 weeks, we see over an order of magnitude increase in antibiotic resistance. So in response to these chemicals, we're changing potentially the composition of the microbial community, but also the phenotype. What are these guys doing? And they are developing antibiotic resistance. So my conclusions about environmental microbes are that the drugs and chemicals that we create for our use, our medicines and our personal care products, are found outside of their intended environment. So not just in your toothpaste, not just in your medicine cabinet, but also going down the drain. Then we find them in lakes and rivers and in the soil and in all kinds of places. And the bacteria in the environment, much like the bacteria in the human microbiome, are going to react by developing antibiotic resistance. So they see these chemicals and they have a way to protect themselves. And they react just as if, just as if this were a regular attack. And because we give them a long exposure period with low chemical concentrations, they actually have a lot of time to develop antibiotic resistance. So we've talked about antibiotics and what happens to them when we take them as drugs and we use them as personal care products. We've seen that they make their way into the outdoor environment by going through wastewater treatment plants. But a funny thing is that they actually wind up indoors and can affect indoor microbiology. So indoor microbiology is studying the bacteria and the fungi that actually live inside the built environment. And the first thing that we have to accept here is that the built environment is far from sterile. So we as hosts, for example, are bringing bacteria in and fungi, bringing them into the environment with us all the time. So we've got bacteria on our skin, fungi on our skin, in our gut, and we're shedding these microbes. Everywhere you go, you shed your microbes. And we try to curb the spread of these microbes, at least the ones that we don't want spread around, by practicing good hygiene, washing our hands, for example. But in addition to that, we also use chemical controls, such as antimicrobials, to limit, again, the spread of what we think are bad microbes, pathogens. And these chemicals we use are not just in things like antibacterial hand soaps or hand sanitizers, but they're also embedded in all kinds of products like these pencils here. These world's best pencils are protected by Microban, and Microban is the trade name of Triclosan. So the paint that's around these pencils is embedded with Triclosan. And we also find antimicrobials in building materials, so things like paints and floorings. Basically, lots of things all throughout the built environment contain antimicrobial chemicals. And lo and behold, Triclosan is then found in indoor dust. So a couple of surveys have looked at various places throughout the world for Triclosan in household dust. And you can see that there's almost 100% detection. That means every sample we take, every dust sample we take, we find Triclosan in there. And we find it at concentrations that are on the low end of what we find in wastewater treatment plants. So this is kind of shocking because wastewater treatment plants are one of the first places that we identified Triclosan as a contaminant of concern. And it's not just in your wastewater treatment plant, and it's not just in the soil outside, it's actually in the dust in your home. So one thing about the indoor microbiome is that it's shaped by architectural design. So the outdoor microbiome, outdoor microbiology is all of the bacteria that live there and have lived there for billions of years. The microbes that colonize us, our human microbiomes, are the ones that are uniquely adapted to grow with us, to either be commensals or pathogens. But the indoor microbiome is a funny combination of both of these things, and we can alter the composition of the indoor microbiome through changes in architectural design and building operation. So one of the things that we're looking at here, for example, is where the air supply comes from. So if we have mechanically supplied air, so that's going to be your HVAC system, your air conditioning, you have a different microbial community than if you have air coming in from the outdoors, from windows, unfiltered air, just an open window blowing in. So it's just one interesting way that we can affect the indoor microbial community. So if we know that architecture affects microbes and we know that chemicals affect microbes, what happens when we put all of these things together? We now have the same chemicals that we are ingesting as drugs or using as personal care products, the same chemicals that are causing antibiotic resistance in the outdoor environment, and now we have the same microbes, but they're all mixed together. So what's happening here? Well, it turns out that just by being inside, the built environment poses a risk of antibiotic-resistant infection. So we know this. We've seen that there are more cultivable isolates of Staphylococcus aureus inside than outside of the home. So if we remember that the blue guys are regular Staph aureus and the guys with the capes are the antibiotic-resistant Staph aureus, and we take air samples from inside and just outside of a home, we find more antibiotic-resistant Staph aureus inside than outside, and these are real data. I've just animated them to be more friendly. We also know from anecdotal evidence that antibiotic-resistant infections, yes, of course, they can be transmitted from person to person, but we also know they can be transmitted to domestic animals that can then transmit them back to other hosts. But even in the absence of another host, there's something about the built environment that can retain this antibiotic-resistant infection and then transmit it back to the inhabitants. So this suggests that there are microbes in the built environment that, just like the microbes in your gut and just like the microbes in the outdoor environment, are reacting potentially to the chemicals that we're exposing them to, the same antimicrobial products that we find in drugs, the same ones that we find outside. We find them in dust, we find the bugs in dust. Maybe there's something going on there. So this is something that not a whole lot of people have looked into yet, and it's something that my group is working on right now. So you can see some lovely pictures of us hard at work harvesting microbes and looking for chemicals. And we're looking at to see if there are correlations, the same way we see them outside, between the indoor microbiome and the antimicrobial chemicals, and to see if there are changes in community composition and changes in antibiotic resistance. And I don't have any data to show you right now, I apologize, but I can give you just my preliminary conclusions from a pilot study that we're running. And we find both human-associated and environmental microbes. This is a picture of the building that we're looking at here. You can see that it is ventilated through windows that open, so we would expect to find a lot of environmental microbes, but also human-associated, the ones that are being brought in by the people who use this building for work and as a classroom. We also find low to medium triclosan concentrations. So this is consistent with the idea that because we're bringing in a lot of dirt, we're bringing in a lot of air, things are flowing through. It's a fairly dilute system. It's leaky architecture, the natural ventilation, the windows that open. There's also what we call high infiltration. Even when the windows are closed, there are lots of big gaps, and so outdoor air and outdoor microbes are still getting in. And despite being a dilute system, we still see correlations between the antimicrobial chemicals and the microbial community structure and certain chemicals and certain antibiotic resistance genes. So just like in the human body, and just like in the outdoor environment, in the indoor environment, microbes are reacting to the chemicals that we humans produce. So drugs and chemical products that we intended just for our toothpaste or just for our antibiotic drugs are found not just in the outdoor environment, but also in the indoor environment. And bacteria in the indoor environment, much like bacteria in the human microbiome and in the outdoor environment, likely undergo changes in community structure and function. They likely adapt in a direct response to the chemicals that we as humans are putting there. So to summarize all of this, just remember that anthropogenic chemicals, the chemicals that we humans make, exist beyond their intended lifespan and outside of their intended milieu. They're not just where we intend them to go, but they go out into the environment. They wind up in our homes, in our offices. And these anthropogenic chemicals are changing the chemical environment. So in addition to the glucose and acetate and all of those things that bacteria are used to seeing, they're now also seeing gentamicin, and they're also seeing triclosan. And microbes react to anthropogenic chemicals, changing the structure and function of microbial communities. So because they see these chemicals, some bacteria are going to survive, some are going to die, some go into a persister state, and some develop antibiotic resistance. So with that, I'd like to thank my research team who helped me think about these things a lot. The collaborators at Arizona State University and at the Harvard School of Public Health. And I will take any questions.
Okay, Erica, thank you for that presentation. I just want to remind our audience to submit your questions via the green Q&A button at the lower left, and we'll try to get to all of you. But if not, we'll be sure to follow up via email. And our first question comes from Russell at Pasteur-Vex Design Corp. Great presentation, Erica. Thank you very much. My question relates to soaps. Are there any commercially available soaps that are used for cleaning yet are not antibacterial?
Yes, absolutely. So most soaps that are not labeled as being antibacterial don't contain antibacterial chemicals. And you can actually see on a lot of the ingredients lists, if you look on the back of the soap, you'll see Triclosan listed, or you'll see, if it's bar soap, triclocarban. And actually a lot of these antimicrobials are being phased out of consumer products. So yeah, look at the ingredients list of soaps and of toothpaste especially. Those are the two big things that we find triclosan in. And there are plenty that don't have triclosan.
Great. That's good to know. I'm going to have to start reading my labels and looking around at my dust, making sure that it's not looking too dangerous. All right. I don't see any more questions coming in. I want to give our audience a little bit of time to just manage the questions. And nope, I don't see any. Oh, here we are. We do have a question. From Diane Hargrave, the College of Southern Nevada, are there historically equivalent events where chemicals, either anthropogenic or otherwise, have had a significant effect on the environment?
So that's a really interesting question. I would say yes, and the few examples I can kind of think of off the top of my head would be things like at the beginning of the sort of industrial revolution, when we're making a lot of progress in one sense and maybe not so much in another, we created a lot of interesting chemistry for things like degreasing agents. We start using rockets. We created rocket fuel. And a lot of these chemicals, when we were first producing them, we thought, we were calling them xenobiotic, which means foreign to life. So not antibiotic killing life, but xenobiotic, foreign to life. And we thought for a long time that these chemicals, because they were things that humans had created, they would just sit there forever. But it turns out that given enough time, bacteria actually evolved the ability to use these chemicals as a source of carbon and energy. So we changed the function of certain microbial communities in places where these chemicals were introduced to the environment. So these are going to be, in the US, your Superfund sites, things like that. Those are a little more localized because it's, one chemical spill in one area, whereas antibiotics and antimicrobials are things that are being used worldwide. A natural example, I imagine there have been, this would be things like, maybe if you have a volcano eruption, or a forest fire, you're releasing lots of combustion byproducts. Those are going to be things like dioxins. And the microbial community in the soil then have to adapt to be able to use these dioxins to be able to deal with things like that. Way back in history, when microbes first started releasing oxygen into the atmosphere, they then had to start dealing with the presence of oxygen. So I would say, yes, there's a long, rich history of chemicals influencing the structure and function of microbial communities on various scales, whether that's the local scale at your Superfund site or on a global scale with oxygen in the atmosphere, or somewhere in between with antibiotics and antimicrobials. Absolutely.
Great. We have our next question from Bren Ames of iOpen Outcomes. Have you considered comparing cohorts of people with hypersensitivity to chemicals for differences in their microbiomes?
That's an interesting question. I had not thought of it, but it does seem like an interesting idea and something worth looking into to see if that sensitivity is derived maybe from something specific to their microbiome, if their microbes are creating a certain metabolite, or if they have a microbe that's specifically sensitive to it. That would be interesting, absolutely.
Great. Our next question, Sarah Rosero from Indicasat. What are your recommendations to prevent or decrease the microbe resistance in the home environment?
So this is something that is getting a lot of attention right now as people are becoming increasingly concerned about antibiotic resistance. So my personal recommendations, I'm not sure. So I think what is being recommended officially by the majority of the scientific community is things like we don't need all of these antimicrobials. Use antimicrobials when you need them, not just all the time. So for example, if you are immune compromised, yes, maybe you should use an antimicrobial soap, but if you're not, you probably don't need it. Things like only take antibiotic drugs when you have a bacterial infection, and especially only if it's a serious infection. If it's something that you can clear on your own, maybe you should not take the antibiotics, but that's, you know, something that needs to be discussed with a doctor. And I think the medical field is revising its opinion on how antibiotics are prescribed and used. So my personal recommendations would be, you probably, unless you have a specific reason, don't need antimicrobial products, so keep an eye out for those and keep them out of your home. And yeah, only take antibiotics when you absolutely need to.
Great. So many great questions coming in. I think we have time for one more from Sophia at University of Barcelona. What is the mean time it takes triclosan to degrade in the environment? And is there any way to accelerate this process?
You know, I have an answer to that, sorry. I think, so triclosan's fate in the environment has mostly been studied in aquatic environments. So its half-life in surface water or in a wastewater treatment plant where it's in an aqueous environment can be quite rapid, because it can be degraded by sunlight. But when it's sorbed to soil particles or when it's sorbed to biosolids, it's pretty well protected. So in that case, it's going to last a lot longer. I'm not sure on average what it is, but it would vary between, you know, on the order of minutes to on the order of years, depending on that exact, you know, how much sunlight there is and what else is going on in that environment.
Okay, this actually is our last question from Bren Ames again. Have you examined the influence of surfactants on development of antibiotic resistance or other changes in microbial community characteristics?
So that's really interesting. One of the things that we don't typically do is look at chemicals in combination. And hopefully it's something that we will start doing more. I think a lot of the technology is getting there to be able to look at microbial community responses in response to multiple chemicals at the same time. So surfactants are interesting because a lot of things that are not typically bioavailable, things that are sorbed to soil, for example, will be released by surfactants, and so they become bioavailable. At least that was the thinking for a lot of, for example, oil spills. To clean up oil spills, to make the oil more available for bacteria, they added surfactants. I think in a lot of cases what actually happens is the bacteria just degrade the surfactants because they're easier for them to get. So I'm not really sure what the response is for specifically antimicrobials and antibiotics. It would be interesting to look at, and I do hope to see more studies in the future that look at not just one class of chemicals, but combinations of chemicals on the entire microbial community.
Okay, great questions. With the questions done, I would like to once again thank Dr. Hartmann for her excellent presentation. And Dr. Hartmann, do you have any final comments for us?
No, thanks very much for your attention, and hopefully I answered some of those questions all right, and thanks again.
Great. If we were not able to speak with you directly today, we'll be sure to follow up with you if you have more questions. We want to thank our audience for taking part, as well as thank today's presenters for joining us here at labroots.com, the leading scientific social networking site. For more information, please visit labroots.com. Today's website will be available for on-demand viewing through February 2016. You'll receive an email from LabRoots alerting you when this webcast will be available for replay, and we invite you to forward that email to your colleagues who may have missed today's live event. See you next time. Thank you and goodbye.